I love this question because it's essentially, why I've dedicated my life to this pursuit. And I think that, the really amazing thing about biology is that biological systems span more than 10 orders of magnitude of scale, right? Like you have everything from the atomic level all the way up to whole ecosystems, or at least like meter scale organisms, right? At minimum, we could say. And I think, our we've been fundamentally limited by our ability to interface with biological systems. And like, what do I mean by that? I mean, our ability to control and to measure them. So if you think about, you ask like the arc of humanity, right? Or the arc of history. So really our only way for interfacing with biological systems for a very long time was through breeding, right? That was our only way that we could manipulate the source code of the system, was through selective breeding. And in fact, that's still, one of the ways that we interface with biology today. But like the real revolution that we've seen in the past 6 decades or so has been step by step being able to build interfaces to each new layer, each other layer of the system. So first, with the heroic age of molecular biology, being able to, we unravel genetic code, right? So we get access to DNA, and we understand the central dogma. We understand how to move, how to read and write things at the molecular level, even if it wasn't achieved. And it took a few decades for us to synthesize the first gene, for us to, for recombinant DNA technology to be invented, and so on. And that allowed us to take control of those systems. But now we're like advanced a few decades, we're able to read whole genomes. There's been like a 10 million fold drop in the cost of DNA sequencing. We're starting to converge on being able to write genomes, both at like the oligomer scale as well as engineered genomes with genome engineering technologies, and the our ability to do that are kind of meeting in the middle to rewrite whole genomes. Similarly, we can read and write cells, right? Like we're able to do full transcriptomics, proteomic maps of cells. We can do it in space. We're starting to be able to do it in time, maybe. Similarly, like we're now using those technologies to learn how to control cell state. So we can, use the same kinds of genome engineering technologies repurposed to actually program transcription to achieve a desired cell state. And you see companies popping up that can, differentiate a cell into any tissue, right? We can read and write the behavior of single-celled organisms, right? The whole field of synthetic biology, and being able to program microbes or being able to program immune cells. We're starting to read and write whole organs, right? You have all these advances in organoid engineering, being able to map cell types in tissue, in situ, like in space, being able to understand development, and then being able to program the genomes of organoids that can model the same kind of development, being able to print the cells in certain locations and stick them together in interesting ways. And so like, it's been just this amazing time where in in a very short amount of time, we've been able to build new interfaces at each layer of these biological systems, which is only going to unlock our opportunities to control, as I like to say, like we've really gained access to the source code of the universe, of so many aspects of life, right? Like if you think about the 20th century as getting access to the source code of physics, right? The 21st century is getting access to the source code of biological systems. And the most creative people I know are starting to think about, what does this mean at like even planetary scales? I'm starting to think about how to use biology to do large scale carbon sequestration, or how do you make Mars livable? So I think the consequences are enormous, the opportunities are enormous, and it's just an incredibly exciting time.